Good morning, everyone. Last lecture we all had discussed with the problem with infertility. We have discussed the infertility problem in female as well as infertility problem in the male. There are so many infertility problems, but what is the solution? The solution is ART, that is ART, assisted reproductive technology. There are various methods of art in which the first promising method is the IVF. This is more commonly known as the test tube baby. You might have heard the test tube baby, but actually nowhere test tube is used into the whole procedure. Let us discuss about the technique that IVF. This particular technique was devised by the Patrick Steptoe and Robert Edward in the year 1970 to treat the woman with block oviduct. The first successful IVF was done into the England, and first IVF baby is the Louis Brown in the year 1978. This is the very much blessing for the couple, those who were not able to reproduce the baby. So this particular treatment was very much famous, although the treatment is very costly, means in lakhs the cost goes, but still this particular method is very much helpful for the couple. But as far as the success rate is considered, although it is expensive, the success rate is only 30%, and it further reduced if the woman is 40 plus age. There are various steps into the IVS. First step is the ovarian stimulation with fertility drug. Now, why there is a need to stimulate the ovary? Now, reason behind that, if the ovary is stimulated, ovary will start giving multiple eggs. Now, why multiple egg is required to ensure the success? If we are having the more eggs, so more and more success rate is there. So for that particular reason, the ovaries are stimulated with the fertility drug. Now, what this fertility drug contains? This fertility drug contains the FSH, anti-estrogen, in the early menstrual cycle. Now, this particular thing controls more and more your ova, or you can say the oocytes maturation. If normal cycle will go, we know every month we are having only one ova which matures. But if this ova will not able to fuse or not able to fertilize in this particular procedure, then IVF goes fail. So for that particular reason, they ensure that more and more healthy eggs to be obtained. So for that particular reason, they give this ovarian stimulation drugs, and thereafter the growth of follicle is monitored. Now they monitor growth with various methods. Like best method is the ultrasound scan, which allows the number as well as the size of the follicle to be measured. Blood test can be also done, like estrogen, progesterone, and LH, to help us certain that ovulation is about to occur, because the hormonal level in at the time of ovulation can be a decisive factor to ensure that yes, this is the date of the ovulation. Date of ovulation is very important. Then they give the HCG injection, which can help to control the timing of ovulation very precisely. Now, if ovarian stimulation drug is given, now next step goes to the retrieval of the mature eggs. This is the second step. Now that particular they are ensure, yes, that now the ova is mature. Now thereafter ovulation has been done, so ovarian follicles are mature but not yet ruptured. Now see, this point is important to remember, that ovarian follicles are mature but they have not yet ruptured. The eggs, about 5 to 15 metaphase to oocyte, they will collect, and then thereafter they are going to keep it into the medium. But how to collect the egg from the female ovary? There are two methods. One method is the laparoscopy method. In that, what they do, laparoscope is a small short type of telescope, as well as a needle is given, or you can say inserted from the abdomen. And see, for that particular reason, general anesthesia is given to the female, and after that they are collecting egg with a small this particular procedure. But intra uterine method is also there, that is known as the transvaginal ultrasound aspiration. In that, what they do, they are using the ultrasound guidance, and they are sucking the oocyte using the aspiration pipette, and thereafter vaginal uterus oviduct root egg passes and comes from that particular aspirating pipette. This procedure does not require any type of general anesthesia, but it is a little bit painful method. So pain medication can be given to the female. Now egg is available. What to do next? Next step is fertilization. Point to be remembered, this is going to be in vitro, outside the body. Semen sample will be collected. Now see, this semen sample will be collected 2 hours before only. After that, they will wash that semen to remove the traces of semen, and then collect the sperm. Now sperm washing will remove the traces of the semen, as well as it will help the sperm capacitation. Note down the term capacitation. Capacitation means the process of maturing sperm. When the sperm enters into the female genital tract after the intercourse, this particular sperm follows the path, and inside the fallopian tube it removes the excess outer core. That particular process is known as the sperm maturation or the sperm capacitation. So when outside, if you are doing this, so the sperm capacitation is carried out by the washing, and after capacitation or after washing, only healthiest and most active sperms retained into the sample. About 1 lakh healthy sperm samples are added in each egg about for 6 hours after the collection of egg in a glass stage. And then gametes are incubated at body temperature, that is 37 degrees Celsius. Fertilization of egg is confirmed upon visualization of 2 pronuclei into the following day. Understood? Now sperm is been collected, egg is also been collected, both are transferred into the cultured medium inside the glass tube, or you can say any type of petri dish like uh, this laboratory tubes they are using, and in that they are keeping it for the 6 hours, and after 6 hours 2 pronuclei can be observed in that glass tube. Now see, this particular thing is when 2 pronuclei are observed into the next day, so this particular pre-embryo are cultured. They are allowed to divide to grow for about 2 to 8 cell stage in the culture medium, and then the embryo, which contains approximate 6 to 8 cells, after third day fluid filled cavity in the they form at the 5th day, and some after time placenta, fetal tissue, all this begin to separate. At this particular stage, we have learned the embryo is called as the blastocyst. These embryo are examined with the microscope, and best graded embryos are selected for transfer into the woman's uterus. Such embryo are ready to be transferred to the uterus at any time between 1 and 6 days after the egg retrieval. Now, pre-implantation genetic diagnosis is done. This is optional, but this is very much important to do. It is performed prior to embryo transfer to screen for inherited diseases. Usually, 1 or 2 cells are removed from the developing embryo and tested for different genetic disease. Only and only healthy embryo that do not have any genetic means problem, they for any genetic disease they are selected to transfer to the uterus. Now embryo transfer, last step of the IVF. This is done by taking a drop of culture fluid through the cervix, and they are transferring it into the uterus using a fine plastic tube. Now, point to be remember, in the case of IVF, embryo are transferred into the uterus. A syringe gently squeezes the embryo along with the fluid into the uterus. No anesthesia is required, but mild sedatives can be given. Woman can leave the hospital after a few hours of this particular procedure and can resume normal life, although she is usually advised to rest for a few days if necessary. Successful development continues inside the uterus now. The embryo hatches out from the surrounding zona pellucida. It implants into the lining of the uterus. Pregnancy test is performed after 15 days of this IVF procedure. The larger number of embryo are transferred, means only single cannot be done. More than one, usually three eggs, or you can say three pre-embryos are being transferred to ensure the successful implantation, because implantation is most important. We have learned that the blastocyst implant or attaches itself on the uterus of the female. So for that reason, if one fails, second will be there, third will be there. But sometimes it happens that all three implant successfully and it give rise to the multiple pregnancies. So this is the procedure of IVF. We will talk about the next method, that is the much more better method than IVF, that is the IVM, in vitro maturation. Now see, this is like IVF only, but what's best about this? The IVF, if you will observe the first step, that is ovarian stimulation with fertility drug. Now, this particular thing is not suitable for the women, those who are susceptible for the ovarian hyperstimulation syndrome or the PCOD, because if they are being treated with this ovarian stimulation drug, so they can cause the various problem like already PCOS. So we all know very well that they are having the already stimulated ovaries. So if such drugs are being given to them, they may suffer, especially the women, those who are suffering with the ovarian hyperstimulation syndrome. They suffer from breathlessness, clotting of blood, fluid accumulation in the lungs or abdomen. Often kidney failure may also occur. So the procedure of IVM will be exactly same like IVF, collecting, but only difference is that they are collecting the egg at the earlier stage when they are immature. These eggs, now we know that every month women, when we were discussing about the normal reproductive cycle, that time we have seen at the beginning of the ovary, means menstrual cycle, woman ovary produces many immature eggs. So all these immature eggs are collected, and these eggs are allowed to undergo to the maturation for a day or two into the dish, which is kept inside the incubator. Then thereafter the egg matures and they are fertilized and using the sperm from the male partner, or you can use the donor sperm if the partner sperm are not good. Then embryos are cultured and transferred to the uterus. Similar, all rest step will be the similar to the IVF. Then next is the embryo transfer. Excess embryo from IVF cycle can be frozen and it can be donated to the couple, those who are not able to produce the embryo or any other problems are there. Even the same couple, if their IVF fails, they can use the cryopreserved embryo, and such excess cryopreserved embryo can be used or it can be donated to the other infertile couples. So this is the idea of the embryo transfer. Embryo transfer, may they are going to transfer the embryo with the various method, that is the Intra-Fallopian transfer. This is the technique in which the embryo produced by IVF are transferred into the fallopian tube. Directly embryo. Now see, what we have learned in the process of the IVF, that the particular embryo, they are transferring into the uterus. Now here the embryo are transferring into the fallopian tube. What is the benefit of transferring embryo into the fallopian tube? The benefit is when the embryo are transferred into the fallopian tube, it program begins stimulating the ovary to produce mature follicle, and thereafter this embryo will just follow the normal pathway. It continue divide naturally fallopian tube and then continue their journey into the uterus where they implant into the inner lining. Means the normal cycle which happens in the normal pregnancy, it begins from the how it begins, how normal pregnancy occurs into the fallopian tube. The egg get fertilized and thereafter it is going to descend into the uterus. So same happen if the embryo are transferred into the fallopian tube. Now next step is the intrauterine transfer, that is IUT. Embryo are directly transferred to the uterus bypassing the fallopian tube stage. They are just simply implanted into the uterus. They perform after the egg have been collected and fertilized into the laboratory. Ideally, between one to three embryos of best quality are selected and transferred into the woman's uterus. In some cases, blastocysts are transferred after 5 to 6 days of fertilization. Now, see, what is the problem with this? Embryo must successfully attach if the wall of the womb for a wife is proper. So, if the embryo which were grown into the laboratory environment, if it adapts itself into the uterine environment, it attaches itself and it shows the successful or viable pregnancy test. Now next is the GIFT technique, that is the Gamete intra-fallopian transfer. GIFT can be asked for this MCQ. Many other questions. The whole infertility treatment as well as ART technique is very important MCQ point of view. Sometimes what happens, students IVF fail because of fertilization and further development of embryo into the artificial environment. GIFT is little bit variation. This is popularized by the Ricardo H from the USA. Now, what is happening in the case of GIFT? Both gametes are transferred into the woman's oviduct and fertilization take place within the body. This is the main difference. Now see, both the gametes means your ova as well as sperm, both are transferred inside the fallopian tube, and it was developed overcome low successive rate of IVF by, because in the case of IVF, what happened, that baby is growing up to 6 days inside the artificial environment, and thereafter this pre-embryo is implanted into the female body. So here in the case of GIFT, it is not happening. And if we will compare, it is less expensive as compared to the IVF. Sometimes the next technique is the ZIFT, that is Zygote intrafallopian transfer. In this case, this particular technique is designed by the Paul Debroy. In this case, what they are doing, they are using the zygote. Zygote means the fertilized egg. Now, here woman's own egg can be used, or if she is not able to produce fertile egg, donor oocyte can be used. Advantage of this technique over GIFT is that fertilization is confirmed, and thereafter this fertilized egg is transferred inside the woman's fallopian tube. The next technique is ICSI, that is intracytoplasmic injection with ejaculated sperm, and it may be from the TESE. Now see, intracytoplasmic sperm injection, it is actually used, or you can say this is the treatment for the male fertility. What happened in this particular case, the male ejaculated sperm are being used. This technique was introduced in the year 1992 for the first time. what happens, some male partner, they have very few viable sperm. So maybe the case is oligozoospermia. We have discussed all these male related problems. So what is happening, they are collecting the good quality sperm of that low sperm count from the ejaculation, and thereafter that good sperms are inserted directly inside the body of the female, maybe the gift and other process. Then thereafter, if success rate is, if you will count, is approximate 25%. The next method is the testicular sperm extraction, that is TESE. Now, this is the treatment for the male, those who are not able to ejaculate, means they may have the erectile problem or some other problem that their testis is not able to ejaculate the sperm. So in that particular case, testicular sperms have been extracted. Now, how testicular sperms have been ejaculated? They, by the process of the biopsy, a small part of the testis has been removed, and from that the sperm is been taken. But now here, the testicular sperms are immature, so the developing baby may have some genetic defect or maybe the abnormalities. The next technique is the PROST, that is pronuclear stage transfer. This is the technique which involves the in vitro fertilization of oocyte, which initially contain two pronuclei. Now here this is something, you can say the modified version of IVF only. They are just checking that the two nucleus are fused, okay, male and female, and thereafter embryo transfer. This technique is very good for the couples, those who have the problem with the oligozoospermia or asthenozoospermia. So these are the various ART techniques which are helping to the couples, those who are not having baby and they are fighting with the fertility problem. But there are many ethical considerations regarding art, like some are saying that it is not right, it is unnatural human intervention. So there are many religious objections made on the moral ground. People are saying this is not good and all. Sometimes some techniques, just now we have discussed about ICSI, immature sperms are used, so it may cause the genetic abnormality and maybe the developing baby is not good, means maybe immature. Many other issues are there, ethical and moral concerns are there, that this pre-embryo transferring and preservation of the pre-embryo cryopreservation, many type of objection raised on this particular technique, but still many people go for ART, and many couples, those who are fighting with the infertility, they are blessed with babies with this technique. So at this note, for today's lecture, we will stop now.